Welcome to all. In this class, we see about the properties of amino acid. First, we see about the physical properties. The amino acids explore some of the properties in a physical solution and the environment. Some are as follows. First, on physical properties, they are colorless, crystalline substances, generally soluble in water. Optical properties next. All naturally occurring amino acid are optically active except glycine, which is optically inactive. So, other than glycine, all the amino acid can explore the optical activity at pH 7. So majority, I think the 19 amino acid are under L configuration, which means the amino acid in a solution that can rotate the left clockwise direction at pH 7, hence they are known as L alpha amino acid. Next we see about alpha means the carbon atom of central chiral carbon atom bearing the both amino and carboxyl group, so that is called as alpha amino acid. Next character, the ionization of amino acid. The amino acid can ionized in any solution. So due to this property, there are three properties are explored. First one, acid base behavior. Next one, amphoteric properties. Third one is buffering activity. First, we know about that acid base behavior of amino acid. The carbo already we know that the amino acid structure. Amino acid contain two functional groups, carboxyl and amino group. Here, the carboxyl group of amino acid can donate a proton and behave as an acid, and negatively charged anions are produced. Amino group of amino acid can accept the proton, which behave acid as a base, forming positively charged cation. So they are explore both the character acidic and basic. Next character, amphoteric properties of amino acid and the formation of switcher ion at isoelectric pH. Any substance having two-way property are called as amphoteric or ampholyte, which means the any chemical substance have explore two property in same substance, that is under amphoteric or ampholyte. The amino acid have also explored these category too, acid and base. So acid donate to proton and accept proton, so they are considered as ampholytes. Next, switcher ion. Monoamino monocarboxylic acid, that means the neutral amino acid. Neutral means the same, exists in aqueous solution as dipolar molecule or switcher ion, which means they have both positive and a negative charge on the same amino acid. Which alpha is the carboxylic group is ionized and become negatively charged anions are produced, and amino group is protonated, which means the proton accepted to form positively charged cation and release ammonia. Thus, the overall molecule is electrically neutral. The molecular species which contain an equal number of ionizable group of, that means equal number of positive and negative ions of opposite charge, and as a result bear no net charge, are called switcher ion. That is the definition of switcher ion. The molecular species which contain an equal number of ionizable group of opposite charge and as a result bear no net charge are called as switcher ion. Next, we see about ionization of amino acid. This is the diagramical ionization of a amino acid. Here is the switcher ion. In this structure, the ionizable form of amino acid. So the cation acidic pH net charge is +1 and net charge -1. So switcher ionic condition, both positive and negative ions are equal. Then we see about isoelectric pH. When this equal number of positive and negative ions bear a particular pH. Okay, so in any solution that release a equal number of ionic state, that the pH of that solution is known as isoelectric pH. The pH at which amino acid bears no net charge and therefore does not move in an electric field is called isoelectric pH. So in a solution, the same amount of ions, in this condition the ions are not move, so electric field, that time of pH is known as isoelectric pH of particular amino acid. Next we see about buffering action of amino acid. The protein can act as a buffer in ICF. ICF means intracellular fluid and extracellular fluid, because the amino acid having both properties, ampholites, can act as weak acid or weak base. In addition, each of the amino acid, acidic and basic amino acid, contain an ionizable group on its side chain. Thus, both free amino acids and some amino acids present in protein can potentially act as buffers. Histidine, among the 20 amino acids, histidine serves as a best buffer at a physiological pH. So, the protein containing amino acid can act as buffer in our blood. The blood pH is 7.35 to 7.45, not not alter in that level. Okay, it causes so many diseases, alkalosis and acidosis. So they are rectified or compensated by this buffer. Example, the if the HCl, that means the acidification, acidosis in acidosis occur in our blood, the base group of the amino group is act and accept the excess H plus ions from the blood. So they regulate the blood pH. And another, if the alkalosis condition is regulated in as alkalosis condition of blood, the base group, sorry, acid group of amino acid actors, so that is COOH, carboxylic acid, that is dissociates into COO minus and H plus. So the H plus is released, that is proton is mixed in blood, so the pH is regulated. Next, we see about the functions of amino acids, that means the importance of amino acid. First, a formation of proteins. Amino acids are joined each other by peptide bond to form amine proteins and peptides. Next, important, formation of glucose. Glucogenic amino acid can convert into glucose in the body, whereas the ketogenic amino acid can convert it into ketone bodies. Enzyme activity. The SH group of cysteine has an important role in certain enzyme activity. The next, transport and storage form of ammonia. Amino acid glutamine plays an important role in transport and storage of amino nitrogen in the form of ammonia. So transamination and deamination give the ammonia in our blood. Then as it acts as a buffer, the amino acid present in protein can potentially act as buffer. Histidine is the major example. Next, detoxification reaction. Glycine, cysteine, methionine are involved in the detoxification of toxic substances. Then next one, formation of biological important compounds. Specific amino acid can give rise to specific biologically important compounds of body.